Our immune system is constantly protecting us from pretty much dying from pathogens like bacteria, viruses, and fungi. I know that might sound obvious, but at the same time, there are also cases in which the immune system can cause more damage than the pathogen itself. This can be attributed to the idea that certain pathogens can induce excessive responses by our immune system. In these cases, the pathogen and immune system can work together almost synergistically to bring about a wide range of symptoms, from bronchoconstriction to organ failure. This idea of an excessive immune response can be found in dozens of different diseases, including the virus responsible for the current COVID-19 pandemic, known as SARS CoV-2. When inhaled, the SARS CoV-2 virus is capable of infecting the epithelial cells that line the inside of our lungs, as these cells express the receptor necessary for viral entry. This virus has been shown to be capable of inducing an excessive immune response, causing immune cells to release many harmful inflammatory mediators and immune signaling molecules called cytokines. In a SARS CoV-2 infection, these cytokines are a method of communication for the immune system and allows for the recruitment of a large number of immune cells to the lungs, as well as to signal for the release of harmful inflammatory mediators. This becomes a problem when the immune system loses control over the release of these cytokines. For example, this can happen when cytokines are initially released from an infected cell, signaling for the recruitment of immune cells to the area, as well as for the release of inflammatory mediators. This can cause tissue damage, and the cells in the damaged tissue will begin to release more cytokines, telling the immune system to become more active, essentially causing a positive feedback loop. This domino effect can result in what has been termed as a cytokine storm. SARS CoV-2 induced cytokine storms can result in significant tissue damage in the lungs in mild cases, and in severe cases can result in multiple organ dysfunction or failure. My research focuses on identifying what cytokines epithelial cells in the lungs are able to release to activate our immune system. I am focusing on a specific class of cytokines, known as alarmins, which are capable of broadly activating several different types of immune cells. Using fluorescently tagged antibodies, we can label intracellular cytokines that are being produced by the lung epithelial cells after being infected with SARS CoV-2. The changes in the cell's intracellular fluorescence represent changes in the cytokine expression and can be quantitatively measured using a software algorithm. Using this technique, I have been able to consistently measure in epithelial cells an increase in an alarmin-type cytokine called TSLP after a 24-hour infection with a SARS CoV-2 virus. With this method, we could potentially identify many cytokines relatively quickly and target them for specific therapeutic treatments to reduce the symptoms of this virus. Thank you.